So thank you for Eija for inviting me here, and I would first like to really acknowledge CSC for organizing this workshop, because there haven't been so many people working with metagenomics in Finland, so now I think there's going to be at least 49 more people working on this topic. So I'm going to first talk of something on how to get genomes from metagenomes, because I saw that that was not covered in the program of this workshop. And then I'm going to tell something of my own results on working with the Arctic metagenomes. I started working with metagenomes in 2009, so after I finished my PhD, and I did my PhD with Petri Auvinen, who was here yesterday from the sequencing lab in Helsinki. And together with him, we did the first metagenomes in Finland, so we were working with the deep biosphere and the food sporage microbial metagenomes. And I really wanted to learn more on the metagenomes, so I went to US to work with Janet Jansen and did some metagenomic and metatranscriptomic and metaproteomic work on permafrost microbial communities. And after that, I've been also working with food microbiology a bit more, and now I'm starting my own group with the Arctic microbiology. So there's going to be more on the Arctic side soon. So you have already learned here is that with metagenomes you can get the microbial community composition, so who is there, what kind of microbes there are in your sample. And then also what is the more interesting part is the microbial community function, that what those microbes actually can do in the samples. There are other omics than just metagenomics. I wasn't here yesterday, so I don't know how much you've been hearing about metatranscriptomics, metaproteomics, and metabolomics. But if you want to work with the microbial function, then there's these other omic tools that you can utilize for that purpose. And here's a picture from Laura Huck's paper last year showing the phylogenetic tree of life, and that was from last year, and it has already changed a lot regarding the archaeal side. But in here, all the phyla that have the red dot in them are the ones that do not have any isolated representative present or available. And you can see that there's a lot of them in here, here, and here as well. And that most of the phyla we really know and have isolates are from the human pathogens and from human gut, as Rob was already mentioning in the morning. So there's really a need for a good reference database for genome database for these different omics tools. And how you can get the genomes for the database? You can of course cultivate them, but we know that less than 5%, less than 1%, or less than 0.1% of the microbes can be cultured, depending on who is saying that. So that's not really the way to start looking or expanding the databases for genomes. Then there's single-cell genomics, where you sort your single cells using, for example, fluorescent activated cell sorting, and then amplify them and sequence, and then get the genome sequence from the individual single cell. But that also has problems, because you need to amplify the genomes, and that might cause some bias, or will cause some bias. And then also because sequencing can produce the really fragmented genome, so you don't have the complete genome and all the information in that. And then the last way to do that is the genomes from metagenomes, which I'm spending some time now telling you about. So when you try to get genomes out of your metagenomes, you get some information that what kind of species there are in your sample, that what kind of functions those individual species have. Because when you're looking at the whole metagenome, you're looking at the functions that are hosted in the metagenome, but not in the individuals inside the metagenome. And this can be, and you can then extract the genomes from the assembled metagenomes by tools called binning. And if I talk of bins, I mean the draft genomes that you have extracted from the metagenomes. Here's a picture from 2009, where showing how the genomes in a metagenome looks like. So here the different lines are individual metagenomes of different GC%, and then you can just extract the information and then look at the context going to each of these bin. The overview how this goes is that you have first your metagenome, you share the DNA and sequence it and get the individual reads. Then you assemble the individual reads into context, and then you classify the context based on different topics. I will talk of those in a bit. and then you bin them, and then you have the community genomes. So they are not complete but fragmented, but you get information on the individual genomes. The binning of genomes is based on, these are the main ways to bin the genomes, so based on coverage. So you take contigs that have same coverage are likely to origin from the same genome. So here's an example of different genome bins, and for example in this one, in genome bin number 3 that I had, you can see that there are the contigs that have really high coverage and contigs that have much lower coverage. And those were something that I needed to then split into 2 different bins, because from one genome you should have a uniform coverage. Then another way to bin them is to use the tetranucleotide frequency. So looking at so here's a contig, so looking at the contig 4 nucleotides at a time, and then you get the tetranucleotide frequency, and then you can build a matrix and bin them based on that information. And then last, there's GC content. So the species from the same genome should have, or contigs from the same species should have approximately the same GC content. Of course, there are problems. For example, 16S can be have a completely different GC content, but overall, that's something you can use there as well. Before I say something of the tools you can use, just a word on the diversity. So this is really easy from the low diversity samples. So for example, Gillian Banfield's group has been working on newborn gut, acid mine drainage, aquifers, and some extreme environments that have only few species. So then it's really easy to get those individual genomes from your metagenome. But if you have soil with, let's say, 10,000 of species or even more, then that's much more trickier. But you still can do it. And then if you want to focus on the rare species, that used to be really hard, because you can only usually get the genomes from those most abundant species that you have a lot in your sample and which assemble well. But there are some tools developed to get the rare species in your sample as well. As for different parts in metagenomics, as assembly and quality filtering and annotation, there are several tools available, and there is not one best tool. But I will show you a few of those tools that you can try with your metagenomes if you feel like it. And these all combine the tetranucleotide frequency and the coverage to get the bins or the draft genomes. My favorite is Metabat, and that's only because that was easiest to install on CSC. So when I installed this, I didn't need to call to Kimmo and ask that, can you help me that I cannot install this? But it was really straightforward to install on CSC Taito server. So there the idea is that you have your samples, you sequence them, assemble, and then you bin them based on the tetranucleotide frequency and the abundance. And then you get your genome bins, and then can work more on those bins to understand the metabolism of those species. Another widely used tool is called ESOM, so the Emergent Self-Organizing Maps, where also tetranucleotide frequency and abundance, or both, are used. So in here, each of these small dots presents one contig, and if they cluster together, then they are, or can be thought to be originating from the same species. So then you can get these contigs from here and then analyze those species. Here's a really nice reference for this tool if you want to take a look at that in more detail. Then there's a tool called Group M, which requires 3 or more samples. So it really needs to have a mass of metagenomic sequence to work. And there is also a requirement that it needs a sequencing platform that has been developed after 1984. And I'm so young that I don't really actually know that what was the sequencing platform before 1984. But I guess there was, it must have been just viewing those, something else than Sanger, but just viewing the chromatograms. So in there, you do the co-assembly of 3 or more samples. And then you need to map the reads back to contigs. Was that something you already did yesterday, mapping the reads? No, not yet. Yeah, okay, so it's going to come later on. But then you know the coverage of your contigs after you map the reads back to them. And then you get the bins. And then you can recruit even more contigs to the bins once you have the draft bins available. And then you can work with your genomes. And then there's also a tool called Concot, which is clustering contigs with coverage and composition. And it uses a bit more sophisticated method to get the bins. So it's also based on tetranucleotide frequency and average coverage, but then it's also using statistical tools to really get the nice bins, which are marked here in different colors. And it also needs multiple samples to work. So once you have your bins, so you have done your assembly and used some of these tools to get the bins, how you can then check the quality of the bins or your draft genomes is to check that they have the even coverage, that then you should use some list of known single copy genes, because each of your bins should have only one copy of those single copy genes. If there's more, then you know that you have a chimeric bin that has more than one species in it. And then what you can do is to do this manually or use different tools that are available for checking the genome completeness and contamination. And my favorite for this is called CheckM. It was also easy to install on Taito, and there you get nice information on the presence of single copy genes and then the genome completeness. And then also you should check the taxonomy of your bins. So either try to find 16 rRNA in your bin. 16 RNA doesn't always assemble really nicely because it's available in multiple copies. But if you find it in your bin, check that you only have 16S from one species, and then use other tools to assign taxonomy to all of your contigs. So what we can learn from these genomes, from metagenomes, is, for example, I took this one because we have the Norwegian people in here. So this was a really recent paper where a completely new phylum was found in the archaeal clade called the Asgard archaea, that otherwise without binning, I mean, these were something that have not been cultured and that are hard to culture, but with extracting genomes from metagenomes, the authors actually found this whole new superphylum and showed that the eukarya actually are originating from the archaea. And then in the paper, what they did there is that they had first their samples, then they sequenced their metagenomes, assembled them, and then they used various different tools for making the bins. So not using only one tool, but they actually used 4 different tools to get the genomes from their metagenomes. And after that, they could do different comparative genomics. So not comparative metagenomics, but comparative genomics on their samples. From the really simple communities, oh, and I'm sorry, you cannot really see my figure was too big here. From the simple communities, there has been study. Here's one example where the authors got thousands of microbial genomes from their low diversity sample, and they could actually then show different ways of carbon, sulfur, and nitrogen usage in the tree of life. So there's various things that you can then do with those individual genomes. And then you can also find novel ways of metabolism to understand that how microbes survive in some extreme environments, or how they, for example, what they showed in here is utilize carbon. So this was a really short introduction to this. I think we could just take the questions later at the end of my talk, because I've combined these 2 talks to one. So now I will change gears and talk of Arctic metagenomes, and I will focus on utilizing those metagenomes as a scaffold for understanding metatranscriptomic and metaproteomic data. So you have heard the word database or often during this, or reference genomes and reference database. So I was having soil samples, so that was not so straightforward for me at first to understand these samples. So I'm not going to talk about anything of databases or anything computational, but just biology on permafrost metagenomes. So at first, what permafrost is, here is Finland, and all the purple here is permafrost regions. And permafrost is a soil that is at or below the freezing point of water for 2 or more years in a row. And you can see that we here in Scandinavia have some permafrost regions, but most of the permafrost is located here in Siberia, in Alaska, and in the Canadian high Arctic. And the Arctic regions shown here with the purple colors actually store over 50% of the global below ground carbon. So that's a lot. And the structure of permafrost is like this, that there is the permafrost, which is the frozen soil, underneath the so-called active layer. And this active layer is a layer that goes through the annual freeze-thaw cycle. So in the summer it freezes, and in the winter it's frozen again. So there's a lot of stress for the microbes in here. And in recent years we have seen that the active layer depth has actually increased as the climate is warming. The summers are warmer and longer, and the winters are not as rough as previously. So it has really increased. And then the carbon here from permafrost is then released for the microbes to utilize. Based on several measurements, the carbon dioxide emissions are increasing as well as the methane emissions, because there's a lot of moisture in permafrost, and when it thaws, all the moisture just doesn't evaporate to the atmosphere but stays on the ground. So they are really moist and methanogenic often. So as I said, permafrost contains over half of the below ground carbon. So we are really interested in that. But what happens to the carbon as the climate changes? That is the carbon then, released to the biosphere, or then are the microbes keeping all the carbon in them and utilizing it for their metabolism? This is a paper from 2011 where they really showed that the climate is warming in the northern regions. So this is north of 60 degrees. So the temperature has been rising there, and based on their estimations is rising rapidly, which then causes the active layer thickness to decrease. So here's the active layer thickness in the late 90s, and here it's what is the prediction at the end of this century. So you can see that it's really getting deeper. And as climate warming is not a Chinese I cannot pronounce it, conspiracy, that there's a data from 2016 from both NASA and from NOAA supporting the same phenomenon, that the climate is really warming. So these are not estimations, but are actually measured data showing that the climate is warming. So in my study, I had samples from permafrost, from the active layer above permafrost, and then a thermokarst bog. So this used to be permafrost area, but as the climate was warming, then the permafrost collapsed and formed this really moist sphagnum bog. And this was the study was published in 2015, and I had 12 samples. So that really shows that how the number of samples you need, or you can sequence with a really small cost, has increased, because then it was a really expensive study and we sampled a lot and sequenced a lot, but there was only 12 samples. But I guess nowadays you need at least 120 samples to publish something in a nice journal. But I extracted the DNA, RNA, and proteins from these soil cores that I had that looked like this. I didn't need to store them in glycerol because they were already frozen. So I just needed to saw them to smaller pieces and then freeze those samples. And then my collaborator measured anaerobic decomposition, methanogenesis, methane oxidation, and iron and sulfate reduction from the same soil cores. So here's a picture how the sphagnum bog looked like. So it was really collapsed. So here's the soil layer as it used to be, and you can see that it has really collapsed and dropped. And this was so that if you would walk there, you would be up to your knees in the water. So it was really moist and really methanogenic. And the trees in here, you can see that they're not straight, but they're sort of, it's called drunken trees. So it looks like they've been having a few beers too much last night. So it's because this frozen soil is no longer supporting their roots, so then they're getting really crooked. So I wanted to understand that what is the community composition and function in the permafrost in the active layer and in the thermokarst bog, and what kind of microbes and processes are involved in carbon and nitrogen cycle and all the other cycles of interest. And then I also wanted to understand that what happens when the permafrost thaws, that how does the community composition, the gas fluxes, and the microbial activity change. And my interest was really in the microbial activity because I didn't want to study the DNA of those bacteria that have been frozen for tens of thousands of years, but really understand that what they are doing, that what is their activity at this moment. And then also to understand that how microbes tolerate those extreme conditions, that what they are actually doing in those, how they are surviving in the extreme conditions. There were some challenges for this study that were at first not computational. At first it was just to get the samples and get the samples frozen back to the lab where I was working at. And then the contamination risks, because I was working with the deep, some people say that it's for the people at VTT, it's not deep, but it was 1 meter depth sample, so I just needed to make sure that I was actually sampling the correct depth and not contaminating the deeper soil layers. The DNA and RNA yields in permafrost were not that high, so I needed to extract a lot of DNA and RNA to get enough for sequencing. And then this is a bit older story, so I was having a lot of computational challenges at first because I was not working only with gigabases but closer to terabases of data. So there was a lot of terabytes of data. And then because it's soil, so it's really diverse. And I was doing a lot of work with the metagenomic assembly to get nice assemblies from these soils. And then there also was only few reference genomes from soils available. I was using several different omics. So at first I did the microbiome, so just analyzed the 16S rRNA from the samples. And what I just want to point out in here is that the 3 different layers I studied, the bog, active layer, and permafrost, were really different in their species composition. So this is now on a phyla level, but you can see that in bog there were those metanobacteria and metanogens as well. In active layer there was mostly actinobacteria and proteobacteria. And in permafrost, the green bar in here, which is chloroflexi, really increased. And I was really interested in the chloroflexi, and especially after I plotted the individual OTUs based on their relative abundance, I could see that there was one individual chloroflexi OTU that was highly abundant in all of my permafrost samples, as well as there were some delta proteobacteria there as well. At that point, there was no reference genome for that chloroflexi. Now there is one sequence from that phylum, so it seems to be really hard then to culture or to catch. But there was no reference genome for that. But I found that it was frequently found in Arctic soils. And I was just wondering that if it's trapped there, or is it really important that would that species give me some information on the microbial survival in frozen conditions, because those mechanisms are still unknown. After 16S, I did metagenomes, so I sequenced the metagenomes for 6 samples. So that was really, it was a lot then, but really little at the moment. So I had 2 samples from permafrost, 2 from active layer, and 2 from bog, and I sequenced this with Illumina HiSeq. I did assembly, but I could only assemble from 20 to 60% of the reads because I didn't want to throw my data out. I was working both with the raw single reads and with the assembled contigs. And my contigs I annotated at IMGM, so at the Joint Genome Institute. So how the metagenomes look like? So this was the DNA part. So in here I have pulled my samples. So here in the tree plot, here's the thermokarst bog, here's permafrost, and here's the active layer. And if the dot is closer to this end, then it's more abundant in the active layer. If it's closer to here, then in bog, and if it's closer to this edge, then it's more abundant in the permafrost. And what you can see in here is that most of the genes annotated from the raw reads actually were shared among the 3 sites. So based on the DNA level, there was not much differences in the communities. What was a bit different was that the green ones in here that are from methanogenesis were more abundant in the thermokarst bog, which I could just say by eyeballing the site. In the active layer, there was more methane oxidation, which is nice that the active layer is then oxidizing the methane, so it's not released to the atmosphere. And then in permafrost, there was more the blue ones, which is sulfate reduction, and then also the red ones, which is denitrification. But that was just the DNA, and I saw that there was not that much differences among the samples. So then I wanted to do the metatranscriptome, so sequence the total RNA from these sites to understand that what kind of genes the species in my sample are expressing at the sampling point. That was done also with Illumina HiSeq, and I sequenced the total RNA. So often when people are doing metatranscriptomics, they remove the rRNA fraction by different depletion techniques. But because I had so little of RNA, and every time I tried to do something with the RNA, then it just vanished because the permafrost RNA for some reason couldn't be heated at all. And most of the depletion techniques require heating. So then I just decided to sequence it all. And the abundance and the rRNA fraction was around 75%, so it was a lot, but not too much because I got rather a nice amount of sequences. And then because I sequenced the total RNA, then I could also then pull out the 60ness from my samples and then get the idea of the active species in my samples. I annotated my RNA reads with mgrast and img, and then I also mapped the reads back to the metagenomes. And then the 60ness reads I just blasted to Green Genes database. And so this is a bit same figure as I showed previously. So in here is the book, here's active layer, and here's permafrost. And on the left-hand side, there's the 60ness, then here's the metagenome, and here's metatranscriptome. So in the Thermokar's book, you can see that the metatranscriptome, so the active species based on the 60ness fraction, really differed. So the proteobacteria were more abundant as compared to metagenome. In yellow in here, also the archaea were more, urarchaea were more abundant. I'm sure that these results would be different if I would have the most updated databases, because we know so much more of the archaea nowadays than what we knew when I was doing these the annotations. In active layer, there was not that much differences among the different fractions, just that the proteobacteria were more abundant. But then in permafrost, there were a couple of things that I want to point out to you. First of all, in the 60ness, so in the PCR-based tool, the Bacteroidetes there was around 10% of them. But based on the metagenome and metatranscriptome, so the PCR-free methods, there was much less of them. And also that in the PCR-free method, the active actinobacteria fraction increased. So clearly the primers I was using were not amplifying those two phyla so well. But what I was really happy for is that the chloroflexine seemed to be active, as it was found in almost the same relative abundance as it was in the metagenomes and in the 16S. I did the same plotting for the metatranscriptome, so just looking at the most abundant 16S rRNA, and I found my good friend chloroflexine 31 here again being really active as well as it was abundant. And then also the delta proteobacteria were found active in the permafrost samples. For archaea, I could see that the metanocyta and metanosarcina were the most abundant OTUs, so those were the metanogenic ones, and it was somewhat expectable to see them in here. For the chloroflexi, I wanted them to drive the draft genome construction, so make the genomes from metagenomes, because, like I said, at the moment there was no reference genome available, and I wanted to understand more what is the species doing, because when I have the 16S, I have no idea what the species can do. So I used Metabat for binning, and then the check-M to check the completeness and contamination of those bins. So when I sampled the cluster at the active layer, it seemed to be too diverse because I could only get one bin from the active layer. From permafrost I got 3 bins, and from the bog I got 10 bins, of which 3 were metanogenic archaea and 2 were sulfate and iron reducing chloroflexi. But in the permafrost there was no chloroflexi, although it was so abundant based on the 16S metagenomes and metatranscriptomes. This is really small, so you cannot really see it, but this just shows some results from the check M. So here is active layer sample, and here are the marker gene copies, and then completeness and contamination. And here is permafrost, the 3 draft genomes, and here are the 10 book draft genomes. And it's really small, so I will read it for you. So the completeness was that for the permafrost draft genomes, there was one that was 89% complete, and for the permakarst book, there was one that was, to check it, 88 and 77% complete. So they were really nice genomic bins. And when I was looking at the functions for these, I could see that, yeah, they're here. So they had several different transporters. So they were really utilizing iron, sulfur, phosphorus, ammonia, among others, for their metabolism, or at least they were transporting those inside the cell. Then they had some of in the permafrost acidobacteria, and then the thermokarst book, chloroflexi, had genes for iron reduction and sulfate reduction as well. And then the methanogenic archaea that I found from the thermokarst book had genes for methanogenesis. So when I did the same tree plot for the metatranscriptomes, the image looked really different, or the results looked really different. So most of the active genes were shared among active layer and thermokarst book. So they are just overlapped here on this line. But what I could see in here is that the same thing as was for the metagenomes. So the thermokarst book had the genes for methanogenesis, so that those were the actively expressed genes. The active layer had genes for methane oxidation. Sulfate reduction was found in all sites, also here in permafrost. And then denitrification was found in active layer and bog. Because I had access to metaproteomics pipeline, I decided to do it, so I have all the tools in the kitchen sink and do metaproteomics for these samples as well. And I had plenty of samples left still in the freezer. So then I extracted proteins using a method utilizing sonification, boiling, and several cleaning steps, because the permafrost has really high humic acid concentration, so I needed to do several purification steps in order to be able to do the proteomics for it. Then it was run at the Oak Ridge National Lab for mass spectrometry. And then at first I mapped my proteins, peptides, against a database made of 180 soil bacterial genomes. So I took a lot of the genomes I could find in the genome databases and decided to use those as my database. But I could hardly see any proteins. So I knew that I had plenty of proteins because when I was doing the trypsin digestion here after extracting the proteins, I knew that I have a lot of protein in my samples. But I just couldn't find any, I just couldn't annotate the proteins available when using the 180 soil bacteria. So what I did then was that, because I had the matching metagenomes, so I then used those as a database for my samples and got a lot of information for this. So here's just to show what kind of so these are the normalized spectral abundance factors, so the NSF values for proteins identified in my samples. And it's not relative abundance, but it's a bit same as relative abundance. So if you would sum these up, that would be more than 100. But it's sort of relative abundance value. And in permafrost, most of the proteins were identified from the active layer metagenome, or from the bog, or from the matching permafrost metagenome. And only few different species were found, or proteins were found in this species that I had in the database as well. In active layer, the best hits were found in the active layer matching metagenome, and same was for bog, that the matching metagenome really gave me information on these proteins. The only bad thing was that most of the proteins that I found were actually then hypothetical proteins or unidentified proteins, so that didn't really give me much good information on them, but I could see that they were found in the same samples as well. So just to go through these results a bit on the metagenome, metatranscriptome, metaproteome part. So methanogenesis I could find with all these different layers of all these three different omic tools from the thermokarst book. Methane oxidation was found in the active layer. Sulfate reduction was with all the different tools found in the permafrost. So that was really something that the permafrost microbes were utilizing. Denitrification was found in active in bog active layer, bog and active layer, but then it was also found present in the permafrost metagenome. And then in permafrost I could find a lot of cold shock proteins and different genes for glycerol transport, so something that they could that would help them to survive in these frozen conditions. So to conclude this part of my talk, so the community composition really varied in different depths of the permafrost areas, and I would really need better databases to annotate the metagenomes. And I really like the multi-omic strategy I used because I could get information on the genes present there, but also on the activity, which I feel is even more important than to know what is the metabolic potential, but to understand the actual metabolism of these species. And then I could also see that upon the permafrost thaws, the different genes were enriched in the samples. So we really need to understand how the microbes in this permafrost, in the thawing permafrost, are reacting to the climate change, and then somehow to incorporate this to the climate models. But because I could not get any chloroflexide genomes from the permafrost, although they were really, really abundant there, I wanted to try something else because I still had some months left at the Janet's lab. So I decided to go to single cells and try to find if by single cell sorting I could get some information from these species, on the chloroflexide species, and then to really get a better database for my metatranscriptome annotation. I wanted to do single cells because what I didn't say in the beginning is that when you do draft genomes from metagenomes, you can make really chimeric genomes. So you can combine information from closely related strains or species, although it should be originating from the same species. So I decided to check work with the single cells, and I did this with Tanja Voike and Rex Malmström from Joint Genome Institute. And something that really made me want to try this was that a colleague of mine, Olivia Mason, was really successful with just 1 mil, so like this much of ocean water, to get single cells from after the Deepwater Horizon oil spill. So I contacted Tanja at JGI because she had published these single cell genome papers. And she said that soil and similar samples have extremely high failure rates, and often results don't look good if we get any. But I still decided to try this just to see if I could find the chloroflexide, because it was a bit of an obsession for me to find something of the chloroflexide species. So what I did there was that I extracted the cells using Nikodem's centrifugation. So I got the cells from the soil matrix, and then those cells were then cell sorted by flow cytometry, then amplified with MDA and 16S sequenced. And after sorting 348 cells from the permafrost, we could find no correct chloroflexi. Then there was a new sort with 6 times 384 cells, so over several thousands of cells were sorted. And of those, all of these, we could only get 103 bacterial cells or bacterial 16S signatures, and of those 2 were chloroflexi. So that made me really happy at that point. And it really shows that the chloroflexi cells are either really small or really strong. And they can be really, really strong because they've been surviving in those frozen conditions for tens of thousands of years. So just some cell disruption doesn't really destroy them clearly, but we would need something more stronger to get those cells broken. So it was possible to get single cells from permafrost, but if I want to do more of this, I'm really going to work on the cell disruption and DNA extraction, because now the cells were collected to 384 plates, where I don't know how many of you have seen those, but the wells are really, really tiny, so they take only around 150 microliter of fluid in them. And then just doing the extraction there was not based on any bead beating, but it was just chemical extraction. So then I think they would really need some harsh mechanical disruption to get the cells broken and then to get the DNA. Because I just could get only 103 cells really extracted from the thousands I had. And then how to increase the genome size, so there could be use of alternatives to MDA, just to get bigger genomes and more amplified. And then what I'm doing at the moment is doing some co-assemblies to the draft genomes and the single cells, or the metagenomes and the single cells, if I would get nicer genomes that way, but then I could also create some chimeric genomes. And what is something that I'm not going to solve it, but I hope that someone is going to solve is it's a problem with the hypothetical change, that we need better databases, that we would have more information on the species we have, because if 40% of your annotation is hypothetical gene, then you're not really going to do anything with that. Or you can do, but then it would be nicer to have more. So I want to thank Janet Jansen and her group, where I did this work, and then my collaborators in US, the funding agency, and then of course you for listening, and I'm happy to take any questions you have.
Hey, thanks for a really nice talk. Where does the Ralstonia contamination come from? I've also seen it with amplicon sequencing. So is it that the polymerases are produced?
It's polymerases, or then I was just thinking that could it be just the naked DNA that is in the soil, that it's just then sorted with the, but the Ralstonia is often with the different reagents. But all of my PCR controls were clean. So it wasn't anything from the PCR products, but it was something that was in the wells where I sorted the cells. So I was thinking it could be either just the naked DNA that is in the soil when I'm doing the sorting, or then it's somehow in the sorting devices that it stays there, although it should be clean. It was purified several times, but yeah. But I think it could be also, for this case, it was not in the PCR reagents.
So when you did where I've seen it, I've seen some traces of that when I do amplicon sequencing for my negative controls. So you see it both in the extraction controls, and also if I do first PCR and then I use a template of the first PCRs or a product of the first PCRs template in the second PCR, and then I pool a maximum amount of that second PCR, double non-template control into the sequencing to get just something. You don't really see it necessarily in the gel or anyway, but you just pool it to see something. And
when
what
you
see
sequence
is Ralstonia and some other beta proteobacterium that I don't now remember. So I wonder, is it, if it's the organism in which most of the polymerases are produced, or
I think I think not. The polymerases are not. I checked that, but I remember now where they were produced. But at least fusion is not produced. I mean, the Ralstonia should not be there. But recently I haven't seen Ralstonia. But now I've been seeing just halomonas.
Sorry.
So recently I've been seeing halomonas as a contaminant. So that seems to be really tolerating all different conditions. But I don't really know where the Ralstonia is coming from. But the PCR controls were clean. But of course I didn't do it so truly as you did. So I just took the template for PCR and saw that it was not amplifying. But all the
I tried to get the maximum contamination, absolute maximum. That there could be.
But it's just, it seems to be everywhere. But I was first thinking that it's the naked DNA, but of course it could be in the fax device or but it seems to be often in especially in the single cell work, but also in PCR.
Okay.
Okay. Hi, Jenni. So I was wondering, when you have these binary genomes from the metagenomic data, how long the contigs they can be?
I've been using always 1,000 base pairs as a limit, but you could use 2,500 base pairs. Do you mean that which size contigs you should take there?
No, I mean which size of contig you get. When you have the genome, it's not a complete genome, but you probably have several contigs. So which is the longest?
It really depends on your sample. You can have hundreds of thousands or or then just tens of thousands. But it really depends on your first assembly, that when you do the assembly for your metagenome, so if you sort those contigs you get from the metagenome assembly, so they might range depending on what is your cut for the shortest contig, but it might range from 1,000 then to hundreds of thousands of base pairs. Okay. But often what I've seen is that you get all the different sizes, so it's not that one species would always have long contigs and one species would always have short contigs, but it varies a lot among the species. So there's a range in there.
Okay. And one other question is like, if you will do again all these metaomics analysis plus the single cell, how much do you think it would cost? Because
okay, let me think. So you should ask that from Petri Auvinen, that how much would it cost, but I would say just the sequencing would be, well, if I would do, I mean, of course I would now have more samples, so I would put there at least for sequencing 20,000 or 25,000 to get more. But then the proteomics is something I cannot say anything of prices for that, because I didn't see any of the price tags for the metaproteomics part. Yeah. Okay. But that's more expensive then, because you need really long times to run the mass spectrometry. I cannot pronounce that, because then you have there are those really abundant proteins that you see at first, but then there are those rare proteins, and of course when you have metaproteome you want to get the abundant and the rare ones, so then I really needed a long time to run those. So that was expensive, and that's why there was only 6 samples and not more.